Hello, and welcome to the post-approval generics session. My name is Ryan Franke. I'm a Director of Quantitative Clinical Pharmacology at Simulations Plus, and I will be your moderator today. Some quick housekeeping items. Please ensure that your mic is muted. You can either type your question in the chat or Q&A during the speaker's session or raise your hand after the speaker is finished. Further details will be provided by Lindsay in the chat. These sessions are acting as a catch-all for activities that could occur after regulatory approval. This include post-approval requirements such as pediatric investigation plans and subsequent studies, post-approval commitments such as post-marketing surveillance or additional information be explored further but does not preclude approval. Sponsor-driven changes such as post-approval changes to manufacturing process, formulation changes, and competitor-driven changes such as the development of generics. The value of modeling and simulation for post-approval changes of a branded product and the development of generics has some overlap. As shown on the left, a variety of models can be developed to support your NDA or BLA submission, including QSP, PBPK, IVIVC, POP-PK, and PK-PD models. The data axis, which is the yellow curve, includes the information and data sets collected through the drug development program. Models describing the relationships between the data sets are captured in the model axis, which is the green curve. The use of these models can optimize the collection of data and accelerate decisions. The dotted curve represents that combining models and data results in higher confidence in the clinical performance at an earlier time in development. Model-informed drug development aims to inform drug development and regulatory decisions by using population PK, dose exposure response relationships, and biological and statistical models derived from preclinical and clinical data sources. As shown in the figure on the right, for generic drug applications, the bioequivalence, or BE, evidence required could include in vitro, PK, PD, and comparative clinical endpoints. PK is the most commonly used data to establish BE, but when PK-based assessments are not feasible or sufficient, then the demonstration of BE may rely on other types of data. Similar to new drug development, when the data collected is combined with a modeling approach, the confidence in bioequivalence, the dashed green line, can reach a higher point at an earlier time. A concrete example is when a modeling approach corroborates in vitro data in the assessment of bioequivalence. Through the increased confidence level on clinical performance for brands and on bioequivalence for generics, we can see how model-informed drug development benefits your development lifecycle, including post-approval studies for brands and the development of generics. We have some really great talks lined up to further expand on these ideas. First, Oksana Jones, an Associate Director of Pharmacometrics at Simulations Plus, will be discussing how pharmacometrics can be applied during post-approval. Then we have Banu Zolnik, a pharmacologist from FDA, who will be discussing the FDA's perspective on PBPK and biopharmaceutics applications. Following her will be Liang Zhao, Director of the Division of Quantitative Methods and Modeling from FDA, who will be discussing how model-informed drug development and model-integrated evidence can support generics. Next, Gustavo Mendez Lima Santos, General Manager of Medicines and Biological Products from Anvisa, will be discussing the role of modeling and simulations in the approval of generics. Afterwards, we have a couple of posters. Inger Darling, Director of Quantitative Clinical Pharmacology at Simulations Plus, has a recording and will be discussing the use of PBPK for drug-drug interaction predictions. Diane Longo, Senior Scientist at Simulations Plus, will be discussing the use of quantitative systems toxicology for drug-drug interactions. Our sessions will be wrapped up today by Brett Howell, Divisional President at Simulations Plus, who will be discussing the application of DILIsym quantitative systems toxicology to evaluate carcinogenic hazard potential of acetaminophen. And with that, we can conclude the moderator's section. I encourage everyone to leave within the Acel Events system and then rejoin for the next speaker, Oksana Jones, after which we will have Q&A session. Thank you very much.